Welcome everybody. As everyone just comes back from our short break and everyone settles, just want to welcome everyone back. It's the final countdown. We are at the last of our global lecture series and engagement ahead of the International AIDS Society's COVID-19 Prevention Conference that's taking place next week. And with that said, I'm very excited that we are going to be talking about basic science, because a lot of some of the questions that were coming up yesterday were about basic science, with a lot of you asking how things are working and asking some of the questions around virology. And that is exactly what Dr. Samukeliso Dube is going to tell us about in just a few minutes, And before that, I just want to remind you that, yes, I will be sharing the link to all the presentations. We just wanted, we didn't want to preemptively share the link to the presentations before Dr. Samu was to begin her presentation. And also at about 7 o'clock, an hour from now, I will also share the link to the post-lecture series survey, and very interested to hear how you've enjoyed it. I've seen the comments in the chat room and all the feedback, but we'd like to collect just one more feedback. And also remember that this feedback is linked to the 10 free passes into IAS, right? So if you filled in the pre-survey and the post-survey that I will be posting in an hour's time, then you stand a chance of winning one of the 10 free tickets. Free, no conditions, nothing. Just free for you to use, or to, if you win, gift it to someone else. If you already have your own access to the conference, it's an important conference. And as activists and community members and community leaders, it is important that you are also part of that conference and are there as scientists deliberate. As Dr. Busiso Konazi said, that it is not just the responsibility of the scientists to lead in the fight against COVID-19 or the pandemic at large, but it is all our collective efforts. We all have a social contract to ensure that we do indeed end this pandemic in all ways that we can. But you cannot end a pandemic without understanding the disease or how the disease that has caused the pandemic works. And this is why Dr. Dube's presentation is going to do exactly that. It will allow us to think very deeply about what are the scientific implications of COVID-19 and how it works. And just to introduce Dr. Samukeliso Dube, she is a doctor with over 20 years of experience in healthcare, including several senior roles in the delivery and financing of healthcare systems, and the latest, which is at Philips Healthcare as a Medical Counsel for Africa and Business Development Manager for Africa. Dr. Dube also worked as the head of healthcare investments, and she was the Africa director of PATH. It is with so much pleasure that I welcome someone who has been part of the struggle. She was there during HIV, her literacy. She can teach literacy. It seeps out of her veins and out of her pores, and I'm so excited to hear what she's going to share with us today. Dr. Dube, welcome.
Thank you, Aponso. Let me know when you are able now to get my slides.
Yes, Dr. Dube, you can share them now.
Okay, can you see them?
Yes, we can.
Excellent. Excellent. And many thanks, Aponzo, and the African Alliance also, and your partners, not only for organizing this. It has been, I logged onto all the lectures myself, and it has been quite not only informative, I think exciting. I think for me, what's exciting, colleagues, is as activists, we need to know what our story is. We need to know and understand some of the basic issues that are around the things that we are advancing. Without further much ado, I'll try as much as possible to make this as simple as it can, so that when we are doing our advocacy, we can converse with all the professors globally at the IAS, we can engage, we can articulate the issues that concern us. And again, to my fellow speakers, I think it's actually insightful to hear some of the experiences from the corners where you sit. So the agenda for today, obviously, I think when you're starting anything around the natural, the basic science of COVID-19 or the basic science of SARS-CoV-2, you need to understand where we're coming from. We always glean insights from epidemiology and history. I am going to cover the basics of virology, viral nomenclature, which actually is important for language. You know, using the right language is very important. A little bit about the structure of the virus and how we detect it, because this has got implications on the testing. And I think the important thing is obviously the life cycle. The life cycle of the virus often allows us to understand why there are certain manifestations at any given time, number one, but also for us to understand where therapies could potentially work. And then the natural history of a disease. What I mean by that is that what would happen, what's the course of a disease when there is no therapeutic intervention. Dr. Mkwananzi was talking about the NPIs, which often they augment some of this natural history of the disease in terms of alleviation of symptoms when somebody is maybe steaming at home, using med lemon, etc. And then obviously from all of these above agendas, we'll talk about the implications for therapy. Where are we now? We are almost in February 2021. We didn't know that our lives would actually be marked by what we are seeing now, with over 90 million confirmed cases of COVID-19 being reported. We've established many things. We've learned a lot. Everyone knows that there is potential person-to-person transmission of a disease, and we've established that. In fact, initially, we didn't know this. I remember last year, this time, when I was trying to understand what was happening in Wuhan, I was worried that you've got a disease which you can transmit when you're asymptomatic. And I was like, this is going to be a game changer. And I went back actually to the notes that were written some time ago by the Bill and Melinda Gates Foundation, where you were saying, you know, we need to prepare for an epidemic, even though as many of us activists have construed it to mean that, oh, he knew that there is an epidemic coming. And of course, he knew from epidemiology. Person-to-person spread is actually a game changer in how epidemics fare. The precise interval in which an individual actually can transmit is an important dynamic in understanding strategies, obviously, that you can transmit before you develop symptoms. And the good news, though, from the data that is emerging and what we are seeing is that transmission after 7 to 10 days of illness is unlikely because of the life cycle that we are going to talk about in the natural history of a disease, especially in immunocompetent individuals. For those who may be immunocompromised, in other words, their ability to deal with the infection, if it's compromised, then the virus may be lingering on for a long time. This, however, is different from when you actually do another PCR test and you still test positive. Because I've seen some people who are testing and testing, they're almost testing every day using PCR, and they are thinking that they are still ill. And the dynamics around testing and whether you're infectious, etc., we need to explain and understand those very well. Because a prolonged viral RNA detection, it may not indicate that you have got prolonged infectiousness. Now, the risk of transmission for this virus from an individual would vary by the type and the duration of exposure. You all know from the history of what we have learned from other infectious diseases, especially such as TB. If a person is talking, it's actually like almost hundreds of of viral particles that actually when you're breathing out. When you are coughing, it's actually more, and when you are sneezing, it goes into millions. So those dynamics around viral transmission are important, because when somebody is symptomatic, when they are coughing and when they are sneezing, obviously the rate of infectiousness is actually higher. Also, the virus is often present in contaminated surfaces. This doesn't mean, though, that this has been the main mode of transmission, especially at home. This has been the main mode of transmission that is significant in places where there is somebody perhaps who is infected, or many people and all the surfaces are actually infected, or in a hospital environment where you've got many people who are positive for SARS-CoV-2, and then your surfaces need to be disinfected now and again. Remember, colleagues, that person-to-person transmission is the most important mode of transmission, hence the NPIs that Dr. Mkwananzi spoke about earlier. Now, overall, the short-term risk of reinfection, however, another dynamic that is important, and when we recall what Nyaras was talking about yesterday when she was trying to decipher the basics of immunology. Overall, short-term risk of reinfection appear to be low. And that may be good news, for herd immunity and our quest, even as we all are trying to get a vaccine. This is not entirely new, even though this specific virus, colleagues, is called novel because its sequencing is different from other coronaviruses. We are seeing, dating back from 1966, that researchers actually found, and this started in the Common Cold unit that was in the UK, where they isolated a coronavirus from a boy who had a pneumonia. In Chicago, they also isolated some of these viruses. And then I think for me, the turning point was in 1968, where they actually emerged this particular virus and gave it a name called corona. And when they did that, obviously they approached nature to say, hooray, we think we've discovered a new virus which is shaped like the solar corona, and we want to classify and name it. Years later, in 2003, this is where we see ourselves battling with the first outbreak that occurred again from southern China in 2003. 2004, some researchers also in the Netherlands in Erasmus Medical Center isolated some of these viruses that were coming from a child with pneumonia, naming it NL63. Another significant finding in Hong Kong in 2005, and then in 2012, where we had MERS isolated again by the researchers in Erasmus Research Center in the Netherlands from a man in Saudi Arabia. This, if you juxtapose this particular trajectory of the discovery of coronaviruses, you will see that what scientists have been doing has been to sequence and understand what are the particles in these viruses that not only confer infection, but that make it possible that this year we've got a different strain of corona, next year we've got another strain. And this is what we have started doing even with SARS-CoV-2. So this is an important one. And in 1931, though, we know that the infectious bronchial virus that was isolated in 1931 sort of was a precursor, and some scientists actually believe that these coronaviruses have been in existence since 1931. Depending on the school of thought that you are at, some believe it's in the 60s, others believe it's way back in 1931. That's not very important, ladies and gentlemen, because we know that we've got a problem in our hands. We've got something that we have discovered actually is has caused economic, social, spiritual havoc amongst everybody, amidst everyone in the world. And right now, in this particular chaos, one thing that I note is that as advocates and as activists, we ought to advocate for more research in science. We ought to advocate for more investments in research, so that even us as Africans, we can articulate what it is that we want to see published elsewhere. No prize for guessing that in all those sequences, except the group that actually did sequence the South African variant of this current virus, we haven't been publishing most of these things. We have not named anything much out from cities in Africa. And I think this virus and this epidemic is showing us where are we investing our money? I digress, ladies and gentlemen. I think the basics of science, we need to understand what exactly is a virus. So in virology, we know that we have to classify these viruses, but what we know is that there are two types. There is one that is naked and there is one that is enveloped. I think if you know that, you would understand that actually it's quite simple. A virus is microscopic. In fact, if you were to compare it with a red blood cell that is circulating in your blood, it's more than 100 times smaller than even a red blood cell. The genome, so every virus obviously will have a genome. What is a genome? A genome is what is encapsulated or what exists and defines this particular organism, right? It could either be an RNA or it would like HIV, or a DNA like the hepatitis B virus, and it could be single or it could be double-stranded. This has got important implications around the life cycle of the particular virus. Every virus often has structural and non-structural proteins that actually surround and form its architecture. Now, proteins on the surface of the viral capsid, in other words, the surrounding, in this particular case, I have put it almost as a pentagon shape. The proteins that surround this actually then are given a name. In this particular case, the enveloped virus, which actually is like the coronavirus, would have this particular capsid surrounded by proteins, and I'm going to discuss more about what these proteins entail. In virology, we often do classification which is dependent on the morphology in terms of the shape. Like for now, we're saying corona because of how the virus is shaped. It's shaped like a crown, and like almost like a solar, the solar corona is actually shaped like a crown. The genome, we actually say, is it an RNA or a DNA virus? The type of replication that it confers, the host and how it behaves when it enters the host, and the type of disease. Recall that the virus itself is SARS-CoV-2. The disease that it causes is COVID-19. So that is an important classification. The important thing, ladies and gentlemen, is that SARS-CoV-2 is a coronavirus belonging to a class of viruses. You've got alpha, beta, gamma, coronaviruses, etc. And it belongs to a class that we call beta coronaviruses. That is a subfamily. So we say order is Nido. The suborder is the Coronavirinae. Family is the Coronaviridae, subfamily orthocorona. I think if you can articulate these, you will do very well at the IAS conference. So the genes or the genera of these is alpha, beta, gamma, delta coronaviruses. And then in most of these actually are in humans, whereas the alpha and the beta are in humans, and they exclusively infect the mammalian species. Others would actually infect birds, like they would infect the avian species. Something to remember, and obviously when you're thinking about this, you almost now know the structure because it has been displayed in various media platforms upon. So I'm sure you always have this in mind when we say corona. You almost can articulate like, what does it look like? We ought to remember. We're remembering this not because it's just fun. We remember this because when we start getting into the realm of therapies, of life cycles, we need to know and understand which hand and eye and nose of the virus actually acts, and why does it act where. And in the space where I sit, where I need to understand whether particular therapies ought to be made available or not, you want us to think about the mechanism of action of these particular drugs. A particular case in point is ivermectin, because I need to understand where does it work, why would it work, and etc. At what stage of the infection should we be giving this particular drug to our patients? That's the nomenclature, and we know that there is a large family of viruses that belong to this group. SARS-CoV-2, we know what it stands for, and is the strain that actually is causing this particular pandemic. The set of symptoms that we see, that is actually COVID-19. What is it that we have? I'm sure now with the vaccine, Nyerazo touched on this yesterday, that when we're talking about the vaccine, everyone is talking about the S protein. Now, what is that S protein? As you can see from this electron micrograph of the vaccine, which is actually it's an animation or a demonstration, the spike glycoprotein, which is the S. Remember, we spoke about the capsid, which is sort of inside and it houses the genome. In other words, the RNA or the DNA of a virus. Then outside there, you've got these spikes, right? We've got a spike protein. Then you've got an M protein. M is for the membrane. So it's in the membrane. And you know, this is weapon. So I wish I actually had a flip chart, right? I wish I had a flip chart and I was drawing all these things. So you've got the spikes, which are outside in this particular ball. Then you've got the M protein, which is in the membrane. And this is important for fusion because we understand that some of the dynamics around how the virus enters the body is dependent upon the configurations that are in this membrane protein, right? And then again, in virology or in science, when you see something that ends with ASE, right? Hemagglutinin esterase, that is an enzyme. So an enzyme, that is the dimer of the particular enzyme. This again, the conformational changes that occur when the virus actually attacks and gets into through a receptor or conforms and joins a receptor are important there, especially these dimers. Then within that membrane, you've got an envelope, and you will see it's often classified as E, because what it does is that it guides the assemble and the release. So when it gets into the cell, it guides that assemble and the release of the genomic material inside your cell, right? And then obviously inside, you've got the RNA viral genome. You will hear vaccinologists and some of the physicians talking about what modality of treatment are you advancing. Some people would be trying to advance whatever is going to inhibit or stop the replication of RNA. Others are going to be advancing something that would stop even entry of this particular virus to my cell, right? And then you've got a lipid envelope as well, which is acquired from the host cell. By the way, the host cell, so my cells would assist the virus in actually its replication or in its life cycle. So I hope that is clear. And Ponzo, please stop me at any given time if I have to repeat this, because I think we're building up to the understanding of the life cycle of the SARS-CoV-2.
Yes, no, definitely, Doc. I'll stop you. And just a reminder to the participants to, if you do want Doc to stop and explain something again, I am watching the chat and Q&A sections, and I will inform her. So please go on. There's nothing so far.
Okay, wonderful. And I hope people are understanding this science, because I love science. I put there to my right a chart that actually depicts what we call it kilodaltons, right? That is a weight of these particular elements of the virus. And we know that as we are targeting the spike protein, I'm saying here it's 150 kilodaltons. What it means is that it's huge, right? Remember, a virus is small, but I'm saying this is actually huge. It makes it kind of difficult for us to find out what's happening inside this thing. We need to understand what's happening inside it so that we can target a vaccine to the specifics that are actually attaching to the receptors that I'm going to talk about later on. So the spike protein is large relative to the other elements of the virus. So just bear that in mind and just be mindful of that. So what are we saying? We're talking about the spike protein. It's going to be important in the life cycle. We're talking about the membrane. We're talking about the envelope. We're talking about the genome, which is actually the RNA of the virus, and the N protein we talk about is the nucleocapsid protein, right? So a virus has got the membrane and envelope, which are the outer layers. And then it's got the inner layers, which is the genome inside the particular capsid. Inside there, you also may have enzymes and other artifacts that aid in its attachment to my cell. What we do in a lab is something that is called sequencing. So when the virus first appeared in Wuhan, we didn't know what this was. We didn't know it was transmitted. In fact, you recall that people were saying, "Is it coming from the bats? Why are people eating bats? Is it coming from pangolins? Where is it coming from?" So what we do in a lab is something that we call phylogenetic analysis, or we can do sequencing. So I can't see who is a participant here, but for those of you who have gone for who have done a paternity test, right? When you do a paternity test, hopefully no one has done that, especially if you're denying your own child, right? You go for a paternity test, you match that DNA, your DNA with your child's DNA, and then you want to see whether there is some relatedness and whether there is actually indeed this is your son, this is or your daughter. This is where the whole sequencing phenomena is actually layered around. In the lab, we also do something which we call sequencing. We want to understand the structure of the virus, but we also want to understand its origins. So it happened in 2020, in 2019, that they wanted to find out whether this is coming from bats, it's coming from pangolins, etc., etc. And how do we establish that it's coming from human to human? Obviously from the epidemiology, but also from what happens in the lab. So that can actually be done. In fact, even in some scenarios or situations, you can take a virus that infects me and the virus that would infect somebody else, and you can match those through phylogenetic analysis to understand whether these are related, whether actually the virus that I have, did I get it from you? So this is something that is often done in the labs, especially for research and for forensic purposes. So a complete genome, it was actually sequenced, and we say it's got a 5 UTR end and a 3 UTR end, and we know that it's got all these open reading frames, etc. The open reading frames 1A, 1B, they are important again in the life cycle of the virus, and I'm going to talk about that later. You don't necessarily need to remember this, but what you need to remember is that in the lab, we do sequencing to understand like what we have done already in South Africa, what was done for the 4 visitors who were coming from Brazil. You know, the P1 strain, we've spoken about the B11 strain that is in the UK. We've spoken about the South Africa strain. There is a California strain that was discussed a few weeks ago. And so with all these mutations, we know that they are different from the wild type virus that actually emanated from China in 2019. So that's what we do in the lab in terms of sequencing. So again, when you hear somebody saying sequencing, this is what they actually come up with. Now, just to sum up these basics of understanding the nomenclature and the virology, structural analysis of the SARS-CoV-2 has revealed through this sequencing that there are genomic similarities but low evolutionary relationship of all the existing SARS. In other words, we have matched this with some of the viruses that we have seen before, especially since 2003, and we know that they are genetically similar, hence the name SARS, right? So they are similar in terms of the genetic sequencing. But there's something that is interesting, that in terms of evolution, in other words, there is the relationship from one generation to another in terms of evolution. Actually, we know that it is also similar to the bat coronavirus in terms of the origin. By the way, globally, we do genetic banking. We put whatever we have sequenced, we put it in a library, so that other countries can reference whatever you find in the lab. There is a library, which is a gene bank, and there is 96% identical genomes between the bat coronavirus and the SARS-CoV-2. So that is an interesting fact, that even though genomically it's similar to the other SARS viruses, but in terms of evolution, it might be that there is a close relatedness with the bat coronavirus vaccine that we have seen from the genus Sape-Covirus. And then there is the important thing about similarities of a genetic sequence is that when you are now getting something that you want to make publicly available as a test, the relatedness should not then confer false positives. And this is something that if you are in a lab or if you are a manufacturer like Abbott, etc., Roche, you need to be very careful about, because implications of somebody testing positive or negative are very, they could be dire. At times they could mean life and death, but at times they could actually mean fear and scrambling around for trying to get resources in environments, especially where we are under-resourced. So that is an important fact. Also, the structural proteins that I spoke about earlier, they are very highly conserved between all coronaviruses. In fact, when this epidemic started, we thought that the coronavirus is not going to mutate as much as it has now, because these structural proteins are highly conserved. But actually, the mutation is coming from these accessory proteins that are generally unique to each of the specific groups of the coronaviruses. So that's where we are seeing the mutations and whatever is different about these coronaviruses. Implications for understanding the structure of the virus, understanding how your body reacts, they are obviously applied in immunology, they are applied in testing. Remember, colleagues, that you were created, right, to resist infections. Otherwise, we would be dead by now, considering all the ho-hos, all the germs, all the foreign bodies that are around us. Your body innately has got some existing mechanisms, whether it's your skin, whether it's your ability to form antibodies, whether it's the cilia in the nose. You've got an innate ability to actually resist microbes that actually infect or they seek to infect you. Now, why is everybody saying we should exercise, we should eat healthy, etc.? We want to make sure that innate immunity, your immune system, is actually strong enough as a first line of defense, because you don't rely on memory cells. You don't rely that I once had this exposure, it's a new virus, so most likely you have not been exposed. So we don't rely on any of that. So we want to build and make sure that our innate immune responses are robust enough, because there are non-specific mechanisms that we rely in. Your cells, your ability to generate an immune response is actually important in the fight against viruses, largely because you know that antiviral therapies is very difficult to come by, those that, especially that would eradicate the viral load compared to bacterial infections. So what we have now at our disposal is the impetus to understand adaptive immunity. And adaptive immunity, if you recall the structure of blood, right? What is blood comprised of? Blood has got plasma and it has got cells. Now, those cells, if you were to divide them further, you've got white blood cells, you've got red blood cells. In fact, I've put some there. You've got eosinophils, you've got neutrophils, you've got basophils, etc. You've got all these types of cells. And white blood cells, so if you were to further divide them, you've got eosinophils, neutrophils, and then the important ones in the cell-mediated immune response are your T lymphocytes. Again, for those of you who are HIV activists, you know that when you talk about target cells, for HIV, we talk about T lymphocytes and specifically CD4 cells. In this particular case, again, we talk about T lymphocytes, which actually should mount a T cell-mediated immune response. David Fred has raised his hand, and maybe you will note that, Ponzo, when I finish this slide. And then you also have the humoral immune response, which is a B lymphocyte response. This is your ability now to generate antibodies. It's mediated by antibodies, and it actually is meant to eliminate extracellular microbes and their toxins. So those are the two types of immune responses. And why I'm talking about this, when you form antibodies and we're talking about testing, remember, an antibody is what you form. An antibody, again, Nyarazo spent some time talking about this. An antibody is what your body actually forms to fight against a disease. An antigen is what this germ, this virus, this bacteria actually then brings, and your antibody, if it's good enough, is going to bind to this particular antigen and form a response. And that response, when we're in the lab, we translate it, whether through an ELISA, an antibody, or an antigen test, and that's where we come up with the result. So in the next few slides, I'm going to talk about testing for the virus so that you understand why do we test and why do we use any particular test. I don't know, Ponzo, whether you want to talk about that question now or later.
We can talk about it right now, Doc. So
Yes.
there was a question earlier on about the structure, but also another question here, is it the COVID-19 has capabilities of mutating? Should we anticipate weaknesses in terms of the vaccine strength?
Yes, so I'm going to talk about that one later on around the issue of mutants. One has to always sequence. In fact, sequencing or understanding the structure, the variant that is circulating at any given time is going to become important for us to curb diseases. We do it all the time. We just are not doing it at this scale because right now we are in a pandemic. We do it for sexually transmitted infections. We are doing it for HIV. We are doing it for many of these infectious diseases. We always sequence to understand what is the strain that is circulating at any given time. And are our antibiotics or our antivirals, are they still sufficient in keeping this? But now this is even more important because we are hoping that we are going to turn the corner around COVID-19 control using the vaccines. So it's important for us to know and understand that these variants or these vaccines that we are having are actually going to respond, and they are still adequate for this particular strain. Now recall that every year people get a flu vaccine, and it's often like updated. It's updated all the time to make sure that it caters for strains that are circulating. So we use maybe what is happening elsewhere to actually detect and predict like what kind of strain is going to come. So definitely you are spot on that understanding the structure of the circulating variant is going to be an important attribute or contributor to us getting a vaccine and being up to date with a vaccine at any given time. The first question was about, again, remind me, Ponzo.
It was about sorry, let me just go to it quickly again. Just hold on, Doc. It was about the last part where you were explaining the structure, the part about the structure. Sizwe was not quite following.
Okay. I don't know, Sizwe, would you allow Sizwe to say what he wants to say so that I pick him up from where he is?
Yes, I will do so in a minute. Just hold on, Sizwe. I also see, while I get Sizwe, I also see another hand that is up, but I think let me get Sizwe first because we are talking about the structures already. Sizwe, you should be able to unmute your mic and ask Dr. Dube the question.
Sizwe, are you online?
He doesn't seem to be unmuting his mic.
Okay, maybe you could continue with another question.
Yes, in the meantime, I see Edith. Edith, you can unmute your mic and ask your question. I don't know if people, Edith, you can unmute your mic and ask the question.
We can see your hand, Edith. Are you able to? Yes, go ahead.
Hi, Edith. Okay, I think maybe she's having issues there with her connection because she's not asking any questions.
Okay, maybe she can write it down,
yes.
I think. Yeah.
There's another question, Doc, and is it possible for someone with strong body immunity to contract COVID-19?
Right now, I don't know what one would mean by strong, right? Because we know that one has to, so strong and strength is very relative, right? Some people say, I'm very healthy, I run and whatever, I'm not going to contract this. In fact, we know some people who have never had flu in their lives, right? And somehow they are able, they mount a very strong or rigorous cell-mediated immune response, and they are able to fend off the virus, or they are able to, maybe they were exposed previously around flu and they are able to do that. But strong is very relative. What we know from science science is that going forward, when you've got the antibodies, for example, against SARS-CoV-2, you will be better off in fighting the virus than somebody who doesn't. And also we have seen from the data that whilst the rate of infectiousness or infectivity is not necessarily reduced, whether you are fit or whatever, you may get it, but recall that 80% of the people may be asymptomatic. So that number, I think, is an important one, because there are some people who may be moving around, they may have COVID, they may not be showing signs and symptoms, but they can transmit. The sad thing about SARS-CoV-2 is that you can still transmit the virus even if you are asymptomatic. So that's why it's answering the question as yes or no is difficult, in that there are many people actually who have got the virus so far, but they never exhibited signs and symptoms, and some actually just exhibited mild symptoms. Only 20% of the people would have overt symptoms, and then that need a hospital admission.
I think Caesar is back. Caesar is back. I'm just going to ask him to unmute. Yes, there he is. Go ahead, Caesar.
Hello. Thank you, Ponso. Thank you, Dr. Samu. I just want to repeat the part of the life cycle of the virus in terms of how does our body help it to replicate. So if you can repeat that part so that I can have an understanding of that part of the life cycle. Thank you.
Okay, thanks, Susan. The good news is that I'm still yet to do the life cycle. So I was doing the structure, even though I was giving some anecdotes and some pointers to the particular life cycle. So I'm still going to do that. And once I go through it, let me know if you still haven't understood. So I've got slides still that are going to talk about the life cycle of the virus. And so within that, it's about cleaning on the structure and saying how then this infers and confers the series of events that we see in the life cycle. Shall I go ahead so that I get to that one, Ponso?
There is two other questions, and I would just think maybe if it's about the part where you were, let's deal with them. Raphael, I've given you permissions. You can ask your question, and then I'll read the others out, Doc, after that. Raphael,
hey,
go ahead.
Hey, good evening. I'm Elvis Raphael from Kenya. Actually, I want to take the doctor back on this topic, whereby like she was facilitating about the I think the signs and symptoms. For me, there was a time I was I went to hospital and I was diagnosed COVID positive, but I didn't have even flu, but it was only headache. Even I never even come across to have any flu, but it was only like I had thorough headache. That was only. So like to me, like COVID has as many symptoms that someone can't even tell. Thank you.
And 100%, Raphael, because the majority of patients, I would say patients, remember one of the important things about infectious diseases is that there is exposure, there is infection, and then there is disease. So many of us are exposed, right, to this virus wherever we go, unless if you have locked yourself into some cocoon, right? We are exposed. What determines whether from exposure you're going to be infected is obviously the barriers that we're creating between ourselves and the next person. Then when you get infected, it doesn't necessarily mean that you have the disease. What is a disease? A disease is when you now start exhibiting signs and symptoms, right? The only challenge that we are now having is that with those who have got infection, even if they are asymptomatic, can pass on the infection. So remember this, exposure does not mean you're infected. Infection does not mean you've got disease. So exposure, infection, disease, and hopefully not death, right? So every time when we're talking about disease control, we actually look at these facets of a disease journey. Limit exposure, If somebody is infected, reduce whatever the infection is going to do in their body, or even infectiousness, so that they do not infect other people. And then disease. This is where we focus on case management to make sure that when you get the disease, you don't succumb. And that's an important trajectory in the control of infectious diseases. And do you have another question, Ponso?
I think the ones from Tariro are directly related to what you're saying right now. What happens to the people that are asymptomatic always, but when tested, they are positive? Does it entail they have a strong immune system to fight something? So just clarity about how a person can test positive when they are asymptomatic.
Yes. So I think that's exactly that exposure infection disease. And now we know that when you are infected, you are likely to have severe disease if you are immunocompromised, if you've got comorbidities like diabetes, hypertension, asthma, etc. And I'm going to talk a little bit about that because of how the virus actually enters cells, and particularly in the lungs, where it actually destroys the alveolar macrophages and the alveolar, the type 2 cells, actually. That's because when it destroys that, and then it triggers other chemicals, cytokines in the body, which then cause damage to all these end organs. And that's where the problem is. So I'm going to talk about that a little bit later. Okay, continuing, shall I go on, Ponso?
Doc, please continue.
Excellent. So now, having understood the components of the virus and why we should test, what can we test, right? You detect the virus presence either directly or indirectly. By direct, what do I mean by direct? Direct is when, if somebody takes my hair, right, and you go and do DNA, you are trying to find out whether it was really Samu or Dr. Dube. You can't do that in the lab. That's direct. That's my DNA. No one else in the world, in the entire universe, can you believe that, has got DNA that is similar to mine. So that is an interesting phenomenon. That's direct. So if you are taking something that is from me and you are saying there is no way this can resemble something else, that's kind of a direct test. But largely these direct tests, in this particular case, it will be your PCR, right? We are doing PCR in South Africa. It is 850 rands. In other countries, it's ranging from $60 to whatever the government or the labs have promulgated. But what is readily available and would be cheaper is indirect methods. Now, the challenge with indirect methods is that the sensitivity and specificity may be lower than the direct test. So we can use, if you can see from this graph, remember I said an antibody is what your body produces to fight against these diseases. So there are types of antibodies that you produce. An interesting and an important angle, right, is that the red line there, it shows immediately when you're infected. Remember, infection is when the virus is in your body, whether you have symptoms or not, but you are infected. So from day one, right, you are going to show the virus. You are going to be positive from the PCR from day one. And this is shown in this red line. In fact, with a PCR, you can be positive before the onset of symptoms. And that is an important one. That's why we are currently using PCR for travel purposes, etc., for ensuring that whoever who has taken the PCR may actually is actually positive because of its high rate of sensitivity and specificity. And in this particular case, we know it's above 96%. What else can we use? I'm talking about the indirect methods. Now, IgA, IgM, and IgG, what are these? These are antibodies. Now, for antibodies, it shows whether you are currently infected or you are recovering from an infection. So can you see that from this graph, when we are doing antibodies, you find that antibodies are likely to come up, or maybe around day 4 or day 5, and then they are going to peak, and then they will go down. And therefore, when they go down, it becomes inaccurate. So from a population perspective, you may not rely on antibody tests for you to effect an effective strategy, or to say to an individual, no, I didn't find IgA, therefore you do not have SARS-CoV-2. So that may be inaccurate. And even other antibodies, depending on what your platform is using, other antibodies even appear later on, like your IgG. And this also has implications on what kind of platform and what kind of testing are you going to deploy for any given population. An antigen test, on the other hand, would be different. So an analyte, so you are saying, what analyte am I going to use? Am I going to use PCR, or am I going to use antigen? And if it's an antigen, what type of antigen am I going to use? And which one at any given point, you have to apply your mind and say, maybe for recovery, I'm going to use IgG because it will show me that this person was exposed, they were infected, and now they are recovering. So can you say that IgG would remain high even after somebody has been exposed? So there is some intelligence in terms of what test one is using. Colleagues, activists, community representatives, etc., please appeal to populations. And I've heard reports of people either buying false tests or they are doctoring tests, etc. This shows lack of understanding of science, because it's for your own good to understand which antibody, which antigen, where exactly do I stand around this. It's not a matter of just getting a certificate. It's a matter of saying, what is it and where do I stand around these particular analytes. And I hope that is clear for everybody. Others even cheat. They would say, I did an antibody test, when they know that it was at the wrong time for them to do the particular antibody test. Summarizing that, when do you use an antigen? Remember, an antigen, again, maybe I didn't explain what an antigen is. An antigen, we say it is that part of the virus. Remember, from the structure of the virus, we said it has got a spike protein, it's got a nucleocapsid, it's got the membrane, it's got an envelope, it's got all these things. So any of those artifacts from the antigen, they are any of those artifacts from the virus, they are called an antigen. So an antigen-based assay is more accurate, obviously, than an antibody-based assay in terms of detecting the presence of the virus. In fact, an antigen-based test would be positive from a few days after onset. In fact, we say between 5 and 7 days, you could use more accurately an antigen for testing the presence of a virus. But unfortunately, it will also weigh down as the viral load starts going down. And remember, the viral load will go down when your body actually effects an immune response, and it actually manages to get rid of most of the viruses. So then an antigen test ceases to be highly specific and sensitive at that particular time. So these sensitivity and specificity tests are actually done over a long period of time, and their applicability in a population setup for testing the presence or the absence of a particular virus. In detecting an asymptomatic infection, again, it depends on the viral load. It may be positive, it may be negative, depending on the viral load. And right now, we're not using it globally, so that's an important one. An antibody, you have seen an antibody, you've got IgA, IgM, IgG, those are types of antibodies. The specificity, not well established globally, but we know that if you have never been infected, it's novel, the virus is novel. So once we have an antibody that is specific to this particular virus, we know for sure that you've actually done what? It's sensitive in picking this particular the presence or the exposure of this particular virus. And I hope this is clear. Now, when we go to the PCR, and there are types of PCR, by the way. The one that we are using now is the real-time PCR. We know you are detecting the gene or the genome of the virus, and it's highly sensitive, it's highly specific. That's why we are sticking with this, for us to be able to control the epidemic or the pandemic globally. Okay, I'm going to just summarize and go through this quickly. You will see there's a ELISA test, what is an antigen in these particular notes, and you can read them afterwards. Primary antibody will bind to an antigen. A secondary antibody will bind to the constant region of the primary antibody. So it goes on, and that's what we use as an ELISA test. So in an ELISA test, we've got a platform, and that platform may have an antigen or an antibody, depending on the generation of that particular ELISA test. And then when you get blood or a swab or a specimen that has got either an antigen or an antibody, it comes here, it binds to the antigen, binds to the antibody, and that strength of binding is what you see, and that's what you read as a result of an ELISA test. Even for a rapid test, that strength, when you see that pink line, is the avidity or the binding, the strength of the binding between an antigen and an antibody that is then reflected in that particular test. Okay, similar to the antibody, again, I've explained the phenomena. Phenomena is almost the same. And if you look here, so the person is the one who makes an antibody. This would be an antibody. The corona would infect a person. Can you see that it has to bind? So when you have got a strong immune response, you're actually having more antibodies that are able to bind the virus so that the virus doesn't cause havoc, especially in end organs like the lungs, etc., etc. And then lastly, the PCR. There are types of PCR tests that we use. There is a reverse transcription PCR, real-time, droplet digital, and isothermal polymerase amplification. But the concept is that I have got a virus, I know its sequence, I get a probe, right? When I get a probe, I actually put it in there, and that probe will attach to this particular sequence in my specimen. And when it does that, it actually replicates. In other words, it amplifies. It makes more of the virions, and that's what I then measure. Currently, we are doing a qualitative PCR, meaning are you positive or are you negative? That is SARS-CoV-2 positive, or are you negative? That is qualitative. Is it there or is it not there? For HIV, you know that we do a viral load, meaning we want to know how many of these virions are there per cubic millimeter of blood. Per milliliter of blood, how many of these virions are there? We don't need to do that right now with SARS-CoV-2, but you will see later on in my slides that there is a correlation between the viral load and the rate of showing symptoms or the severity of disease for a person. So viral load actually has an implication on the severity of disease, but we don't measure it because we are not going to do anything about it. So we apply this knowledge to understand false positive or false negatives, how to collect a specimen. So now I know if I want antibody antigen, where is the best place to collect a specimen, right? Where does the fighting of the virus actually take place more, right? We know that is we collect oropharyngeal because we know that's where the activity takes place. Often when somebody has not collected a specimen properly, you've got problems because you might have a false positive or a false negative. At times there are issues in the lab that may happen. At times there may be mutations, and this is what we are watching against. We are watching in the sequencing and in the library or storage of these particular sequences that we do not have any mutation at any given time. And also when you've got concentrations that are below the threshold that we are measuring, the specimen wouldn't be, even though it's appropriate, it wouldn't have enough of the virions for you to test positive or negative. And when you are new in terms of using this technology, again, you may have false positive or negative. So understanding these procedures is very important, and please just familiarize yourself with when do we use antibody, when do we use antigen, and when do we use a PCR, so that as advocates, we can advocate for availability of these technologies in our vicinities or in our communities with knowledge of what they do in terms of conferring the epidemic. What Sizwe was waiting for is the life cycle of SARS-CoV-2. But before that, Conso, I don't know whether there are any questions.
There are questions, Doc. There are a few questions. Let me just go through them. Marsha is asking, saying, thank you so much for your presentation. I can see that you understand virology very well. I would like to know what makes the coronavirus different from the HI virus and why there is a vaccine to fight one virus and not the other, since both attack cells responsible for our immunity. The second question is from Moses, and he's asking, to what extent is our political leadership well versed with this detail as they are involved in the decision making to open or close borders and to combat the disease, the spread of the disease. So it's asking if our political leaders are, you know, that how much do they know, and are they making informed decisions? Those are the only two questions for now.
Okay, I think both of them are somewhat political, right? But I'll attempt to start with the science. There is obviously many viruses have got this kind of life cycle, that viruses are kind of parasites, even though we don't classify them as parasites, that in that they thrive when they are inside the human body, or when they are inside these cells. So perhaps those are some of the similarities, but vastly we know that the life cycle of HIV, also in terms of integration, etc., is a whole big lecture on its own. So if you're interested in that, we can discuss that later or some other day. But what we know here, and why the speed with which we have developed or we've understood the life cycle, number one, and the speed with which we have actually come together in solidarity globally to develop a vaccine, is also the extent of the impact of this particular virus. We have pivoted from the platforms that were actually being used in developing an HIV vaccine, but the funding, no other disease in history since you and I have been born has had funding like SARS-CoV-2. So with that level of funding, with that level of pulling expertise together, it has been easier to actually come together and develop a vaccine for corona compared to HIV, where I was one of the investigators in one of the IAVI vaccines some time back, and we didn't have like lots of money for us to always continue with our studies, right? But here we know that this is the level of effort, the level of camaraderie actually is unprecedented. And utilizing that, many scientists have pivoted from the platforms that have been already created from discovering and finding an HIV vaccine, and they've used them now. Had we had this epidemic in 2009 or 2008, when we were still struggling around identifying which epitopes, which targets for immunity for HIV were going to be relevant, I don't think we would have done this speed of discovery of vaccines as we have. So technology, whether it's technology for DNA, for RNA extraction, whether it's technology for identifying these epitopes, etc., it has been easier now than it was then. That's number one. And also, when you've got a disease that is infecting large numbers of people, it's easy for you to enroll. And as Nyerazo was saying, you know, when you enroll this large number of people, it's easy for you to get a powered study where you are able to draw conclusions. But in HIV, I remember we would wait, and then you stay a month, you haven't had anyone who you could enroll, and then no one would seroconvert within your study. And so it's difficult for you to easily draw conclusions. So what we have learned is that we need large numbers of people for us to draw conclusions around HIV. The other questions around political leaders. So for every country that is well-meaning in its fight against COVID-19, they would have a command council, right? They would have trusted advisors who understand science and who understand public health, and as Dr. Mkwanans was saying, who understand community issues, who would bring in all facets of life in tackling a pandemic around the table for advice and for generation of policies. It is not possible for one to actually come up with a strategy that works alone. So you need a council of advisors who know their story. So one hopes that all the promulgations around laws and strategies to combat this epidemic have been informed by these advisors. We certainly have one in South Africa. We perhaps need more community representation in that particular council. And then we need to have our voices, everybody's voice, to actually be heard and to be pulled forward so that we can all fight this epidemic. Shall I move on, Ponso?
Yes, Doc, you can move on.
Okay, briefly, colleagues, in terms of the life cycle of the SARS-CoV-2, I've highlighted the structure of the virus. We know that in terms of the first step that happens, there is binding, right? What binds? Just like HIV, we know that there's GP120 binding. Here, what binds? Remember, I spoke about the spike protein. There is spike 1 and spike 2. The spike protein actually would bind to the ACE, which is the angiotensin converting enzyme 2 receptor that is actually in humans. So this is what I have. I have the ACE, ACE2 receptor, and it will bind to the S1. So that is binding. So I hope that is clear. And this is there articulated, or it's actually put in the diagram there, that there is actually the binding. When that binding actually happens, there is a transmembrane protein that is thought to actually be the most effective, that determines virulence. So there is binding, and then there is the cleaving of the S. So spike proteins, there is 1 and 2. There is the cleaving of the S2 protein that then happens, and that one is facilitated. Obviously, for me, I've got this receptor, the TMPRSS2. So you hear about that, which is a transmembrane protein that is there. So there is the binding, and there is the cleaving of the S2, so which is facilitated by this. And that, ladies and gentlemen, confirms the rate of infectiousness of this particular virus, because they are saying that the S1 SE2 binding is really not of any huge consequence, but this second binding, that then with some transformational change, is the one that really facilitates the virus entry. As we are talking about these steps in the viral replication cycle, remember where drugs act. Remember, when we're talking about antiviral agents, most of them, we want them to inhibit or to stop this virus from binding and attaching to my cell, or we want it to stop this conformational change. So if you're a drug discoverer, you are saying, at which point do I want to actually work? So that is step number 1, which is the binding and this transformational change. Now, when the binding then occurs, remember, inside the capsid, there is this genome, right? The genome has to be empty, because that's where the replication, that's the engine of who I am, the engine of who is going to become the next Sizwe, right? It's your DNA. So that's the one that you want to see replicating. So when the entry then happens, it fuses with the viral envelope, it fuses with my membrane or with my endosomes. There's something that we call endosomes, right? Endosomes in my cytoplasm. This is where, Ponzo, I say you should all remember what you learned in grade 5 or grade 6. You learned there is a cell, it's got a nucleus, it's got a cytoplasm, etc. You should remember those things. And there are endosomes. So you should just remember, you're not learning that for mahala. You are learning this for a time such as this, so that you understand the life cycle of SARS-CoV-2. Now, the endosome, binding to the endosome, then it actually creates, these are the caps, the envelopes, and then it will fuse and then uncoat. The uncoating, I say, it leaves ipachi, like the coat, it leaves its coat outside, so that the genomic RNA is the one that then gets into my cell, because that's the important part. It is emptied. Emptying its contents inside is a very important step, obviously, in the replication cycle of the SARS-CoV-2. So that transmembrane protein, I said, is the one that actually facilitates. It's efficient. It's the one that confers the efficiency of the transmission. And then once inside, these viral proteins, they would then translate. Now, in genetics, we talk about translations, transcriptions, etc. Let me simplify this. So when I've got my DNA, or in this case, we're going to start with an RNA. When I've got my DNA, I want whatever is going to be produced from my DNA to actually be familiar, or to be similar to that particular DNA. So how am I going to do that when I just have this one DNA that is only a double helix? So you have to form what? Copies. So you replicate, forming particular copies. So you translate that. So you have to translate that into some RNA, and that RNA then would be translated into the proteins, and the proteins would either be structural or non-structural. So that's the sequence. I need to form a platform that would have these amino acids, and amino acids, when they're in a certain sequence, they then bring out a certain structure. So you've got this RNA, it gets in, and I don't know whether you can see my cursor. So you've got the RNA, the genomic RNA, it goes in there. There is a complex that is formed. So there is a complex that has got this structural protein, and it does viral RNA synthesis. It will be translated now to these amino acids, right, that are going to form all this in the most of the genomic RNA. When this is actually done, they would assemble together, they would move through the endoplasmic reticulum, which is another organelle in the cytoplasm, and then through the Golgi apparatus, and then will move out of the cell in a process called exocytosis. So if we know that once it does that, the cell will die using apoptosis. So the cell actually then dies once this cleaves. Remember, if you are making holes in the cell, right, it's going to then die, and these virions actually are released and they go and infect other cells. So in summary, there is the binding, right? The S2, actually there is the cleavage that happens for S2 to facilitate fusion. And the fusion will take place either at the cell membrane or at the endosomal membrane. That's where the fusion, and once that fusion occurs, we know that the virus is actually inside my cell. Then there is uncoating of the all the coat. I say the coat, removing the coat of the incoming genomic RNA, which is then dumped inside my cell. From the genomic RNA, there is the translation of the two, we call them ORFs, which are the open reading frames. These are translated into polyproteins, right? So you are translating this RNA eventually into proteins. Core and post-translationally, they are processed into individual proteins, right? And then there is transcription. So I always say you translate and then you transcribe. So you translate from RNA to these particular proteins, and then you transcribe. When you're transcribing, you're saying, how should this thing actually look like? So you transcribe these subgenomic mRNAs, and then which actually characteristic nested set of coronaviruses. They are these coronaviruses, which then are translocated into, in other words, they are transported via the endoplasmic reticulum, which is another organelle in the body, and they transit through the Golgi apparatus. And then in a process that is called exocytosis, they are going to then move out of my cell. They are secreted, they go out there, and they go and infect other cells. Is that clear? So majority of this, in terms of where the cells are, you find that this virus loves the cells of the upper respiratory tract. So in the predictions that we had for pandemics, we thought it was going to be lower respiratory tract, but being in the upper respiratory tract, it's a problem because now the natural history of the disease actually changes. If it was lower, it's difficult for you to infect somebody when you've got a lower respiratory tract infection. They love the upper respiratory tract, and this is the problem that we actually have today. Now, in terms of the natural history, just quickly, Ponzo, when I'm saying natural history of a disease, I'm saying how will it fare when there is actually no infection or no therapeutic intervention. Stage 1 is early infection, where the virus actually just replicates, and people would have asymptomatic infections, like you would have maybe flu. They're like, oh, I contracted a flu, I had some mild symptoms, or I didn't have any symptoms. That it doesn't mean that you are not infected. You can detect the virus by a reverse transcriptase PCR. The next stage often is when now it moves from the upper respiratory tract and it gets into the lower respiratory tract, or even other organs later on. When it does that, the affected lungs, in the lungs you have a pneumonia because of the alveolar cells that are there, especially the type 2s. They are the ones that seem to be exhibiting the ACE. Remember I spoke about angiotensin converting enzyme, the ACE. It seems there are many of these cells in the lungs that actually have the same receptor. So once there is a receptor like that, the virus loves that particular receptor. Just like HIV, it loves anything that has got a CD4 receptor. So these ACE receptors, where they are prominent and where there's this transmembrane protein that I spoke about, that's where actually they attack and that's where they divide. And when it gets into the lungs, we know the symptoms, cough, dyspnea, shortness of breath, and actually many people who are hospitalized are in stage 2. And then stage 3 is actually something that was described largely when we were in the first wave for most of our countries. We're talking about the cytokine storm or the hyperinflammatory phase. In other words, remember, inflammation is not a bad thing. Inflammation is a phenomena that tells your body that there's something wrong, do something please, do something please. So it sends signals to the brain. It says, bring all these cytokines, I need these interleukins, I need these interferons, etc., to be able to fight. So this overly excited host inflammatory response, it's actually meant for good, but then it causes damage. For example, some of the interleukins, they make the blood vessels porous, such that some people actually die of clots, of blood clots that are all over. And others like I think it's the interleukin, I think 17 or so, it actually causes healing by fibrosis. So it causes fibrotic lungs. And then when you are trying to heal, you are then actually making things worse because your lungs become rubbery and they are not able to respond properly. So all those syndromes that come from hyperinflammatory response, they are the ones that we see manifesting for our patients who are often hospitalized. The challenge and perhaps even the folly to Dr. Samukeliso's answer earlier, that you hoarding oxygen, you don't know what you're going to come up with. You don't know whether yours is going to be failure from respiratory distress or from kidney failure or from whatever. So you don't know. And I know in some countries where we had politicians hoarding ventilators, etc., you don't know what is going to happen in terms of this particular history of a disease. And therefore, at times, it may be folly for you to just think it's just oxygen, because clearly it's not just oxygen, that the oxygen shortage or hypoxia that might end up killing patients. Again, please take time to read some of these notes in terms of the virus is inhaled, there's local propagation, and then it goes on, propagates down in the respiratory tract, and then later on it causes end organ damage that is often caused by the cytokine storm, or at times is caused by the inflammation, the damage to the blood vessels that then occurs. There is a relationship, and I'm not going to dwell into some of these studies, between the viral load and the severity of disease. So if you have gotten a huge load and you are not able to mount a response, it means that there is a huge viral load which actually causes more damage in your body, and this then is translated to the severe symptoms that we then see later on. And then in terms of the natural history, our understanding in epidemiology of the natural history of a disease is the one that that actually helped us to have some of these policies of wash your hands, cover your mouth, etc. in prevention. That's what is also helping us understand what we can use for a vaccine. And lastly, what is helping us in understanding therapies that are going to work. This is a last set of slides. I think I've got 2 more to go. When we are talking about basic science helps us understand drug discovery. And broadly, we've got, in terms of drug discovery, we've got drugs that stop entry. For example, and I know remdesivir is one of those drugs that is purported to do that, even though the clinical trials are not showing much of its efficacy. You want drugs that may inhibit or stop this virus from replicating inside the human cells. For example, we've got some of the drugs that would stop cleavage, for example. And these therapies, they are there in testing, and I'm going to show you a table later on. You may want therapies that avoid this cytokine storm that I've spoken about, so that you don't have this inflammation. And by the way, why people who have got hypertension, who have got diabetes, etc., would compare worse in terms of disease severity is because they already have ongoing inflammation in their bodies. And when you now add with this cytokine storm and the hyperinflammation, it just becomes worse. That's why comorbidities make somebody worse off in terms of the outcomes than somebody who is healthier and doesn't have comorbidities. The other groups of drugs, you obviously may want to just manage or even salvage organ damage. It could be therapies, it could be support, whether it's oxygen, whether it's renal dialysis, etc. You're trying to manage and stop some of these this support. And then in terms of clinical trials, there are groups of drugs that block entry, block replication, and I've given examples there, like ribavirin, it actually would stop replication. And then there are those which actually boost innate immune response. So far, clinical trials are not showing much promise there, even monoclonal antibodies, and then those that enhance antiviral innate activity. Those are the groups of drugs. And in this particular diagram, which you can actually study a little bit more, it shows antivirals there, you know, your chloroquines and your hydroxychloroquines. They were actually meant to avoid from the membrane fusion. They were meant to actually stop this process from happening. Antivirals, again, stopping viral entry, particularly the cleavage that is caused in the plasma membrane. Those are some of the drugs there. And then this is kind of the favi period, which actually would stop the genomic RNA transcription. This is another drug that is there. Others in this complex would stop even translation from happening. Remember, I spoke about translation. Remdesivir, lopinavir, ritonavir, they were hailed as promising, but as you all know, this is not necessarily what we are recommending broadly. So we've got all these drugs that would work at any given point in the life cycle of the virus. But the news is that of all these, we are not seeing something that we can surely say for sure. We are there. This is a wonder drug. There are no wonder drugs. There isn't something that 100% works. There isn't anything. We just have regimens that we've put together as countries to actually try and ease where we are coming from. It may still be too early for us to really say this is what we can do. Even some of the therapies and supportive agents that are hailed as we ought to do this, we ought to give vitamin C, D, zinc, etc. You'll find that authorities around this, they say, yes, but we don't have conclusive evidence. So evidence gathering is still ongoing. We do not have a wonder drug, including the ivermectin you saw from SAPRA, and I'm seeing some of the regulatory agencies around Africa, they are saying it's going to be available. Recall that we do not have data. We do not have evidence that say for sure, this is a wonder drug. Human beings want a wonder drug. Human beings want a solution, a silver bullet. Currently, ladies and gentlemen, it does not exist. What we can continue to hope for is combination of these therapies in clinical trials coming up with results that we can all at least hail as effective and efficient in stopping the transmission of SARS-CoV-2. So with that, I think I'm going to stop there, Ponzo, and I'm going to take questions.
Thank you so much, Doc, for that incredible presentation again. It is really amazing to just hear all of these presentations and to understand the science and how the science will help empower what is happening, you know, help empower community leaders to ensure that they are able to you know, advance the causes of their communities, and in ways that also allow for that they become the people who understand this and also translate this in their communities. It's very important, and I hope when we think about this, and when we think about what this means particularly in connection to advocating going into IAS. But before we even go into a discussion, Doc, there are a few questions, and I will give you 3 at a time. there's lots of gratitude also towards you for your extensive explanations and simplifications of such complicated scientific knowledge. I don't think, you know, I've heard this much science since I was in high school, but it was so interesting and I learned so much myself. There's a question that says, did the CRISP technology to develop do they use the CRISP technology to develop the SARS-CoV-2 vaccines?
I actually am seeing that on the panel. I don't know what technologies they've been using in that. For me, I think I just understand the targets that they were targeting when they were doing using vaccines. So each company would have different focus areas for the particular basic science. In terms of the technology that then was used, I think we should have asked Nyarato yesterday to explain if there was a platform or a technology that would have been different. Perhaps the guy who asked and is anonymous could write to us around these various technologies that then would be used. So the technical know-how around how you then make the vaccine, it's not my area of expertise, but what I would know is what are you targeting in terms of basic science at any given time.
No, definitely. Thank you so much for that. And Doc, just wanted to also ask, you know, how do you think there is enough translation of science into human or into normal languages for communities such as activists and other communities of people who work in the public health advocacy space?
I personally think there isn't much. Learning from what happened in HIV, I think though there is an opportunity for us to scale that up, right? So for HIV, we had all these web pages where we translate science into something that you understand every day. And not only did it make activists knowledgeable, but it sort of destigmatized HIV. And you find an activist saying, you know what, oh, bring my ARVs, I need to drink them, and they're in a meeting, right? Because they understand the science, they understand everything. I don't think there is enough. In fact, it's not just these diseases, about demystifying science. If you are on Twitter or some of these things, there is the naked scientist. I don't know whether you know that virologist talks about science in a simplified way. I actually love, you know, how he demystifies science. We need more Africans who are going to do that. If we had, we wouldn't be having people talking about 5G and all these conspiracy theories because we don't understand science. I think for me, it's high time Africa not only invests in science, invests in research, but an understanding of research and science actually makes your decision making different than for somebody who doesn't understand.
No, definitely. And this is why this lecture series was so important, particularly to support the communities that will be going into IAS next week for the COVID prevention conference. And Dr. Samu, thank you so much again for, you know, closing it on such a high note. I think we've come full circle. We started yesterday with lots of science on vaccines, and today we're ending on the basic science of the novel coronavirus as it is, what happens when it's in our bodies, and what happens and what we should know about in understanding the way it functions and why we are still in this pandemic. I'd just like to thank all 4 of our speakers and guest lecturers for the past 2 days. It has been incredibly learning experience for myself, and I see for so many of you who've said so in the comment sections on both days. Please, just a reminder to fill in the post-workshop survey. This is to help us know that if we ever do get a chance to do this again, how we could make it better and much more to your liking. So please do fill it in. And remember, if you filled in the one from earlier today and yesterday, and this one and the one that I posted just a few minutes ago, you stand a chance to win 1 of 10 tickets. You know, when you guys log off here, the team and I are still going to look at who those, put it in an electronic raffle and find the winners, so that by Monday we are able to email everyone their passes. But, you know, we started with Dr. Nyaradzo and Dr. Marua, who showed us, who talked about vaccines in particular. You know, they went into explaining, Dr. Nyaradzo was explaining the science behind vaccines, how vaccines are manufactured, answered questions that I've seen many people ask, even on social media, even on the radio. How did we develop a vaccine for COVID-19 so quickly? You know, and it's when the science is explained, when the science is broken down, that we get these answers. And then Dr. Jennifer Marawa went into the politics, the policies, and what is holding Africa back. Why didn't Africa have enough clinical trial participants or clinical trials for the vaccine happening on the continent? What are some of the geopolitical things that are happening in the political world, in the socio-economic factors that leave us, or leave the global south behind? And also just talking about global access and equitable access, and holding pharmaceutical companies also to account in the fight against the COVID-19 pandemic, and in the race for a vaccine. That if patent laws, or if patents are put on life-saving drugs such as a COVID-19 vaccine, then we live in a truly horrible world. And then today we started with Dr. Spusiso Mkhwanazi, who gave us an incredible presentation about non-pharmaceutical interventions and how they work and why they're important, but also the implications it means for inequal societies that many of us live in poor communities. What does it mean for, what does social distancing or isolation or quarantine mean for a family of 10 living in a 2-bedroom house? These are some of the things that, you know, we need to take into consideration. Policymakers need to take into consideration as well. And this is why non-pharmaceutical interventions are so important. And of course, what Dr. Dube has been sharing with us for the past hour, which is also incredible, around the basic science of COVID and COVID-19, or the novel coronavirus, which is, it's very important, as she said, you know, that we need to call the things what they are. So COVID-19 is the disease that is caused by the novel coronavirus. The virus causes the disease that is COVID-19. And also just to make sure that our language and how we understand the science, we are able to also translate it in our communities. From the African Alliance team and all its partners, thank you so much for joining us for these 2 days. We really appreciate everything. Fill in the survey. You will be notified by email. There's also a link to the presentations. You can download them. All 4 of them are available. I'm Bonsa Pilane. Thank you so much for joining us over the past 2 days. Have a good evening. Have a great morning or night, or wherever you are in the world. Thank you.